So I wrote a paper recently looking at if the scientific community decided that it really wanted to have a way to replace organs as soon as possible with as little money as possible, which would be your better choice? Working on developing prosthetics or working on manipulating biology? Now, I'm a big prosthetics nerd, and I've always kind of liked those elements of transhumanism, melding man and machine. But I was really blown away at how much closer we are to being able to repair or enhance our bodies using biology than we are using machines. I'm going to kind of skim over some of the paper that basically can be summarized as both are totally possible and get right to the good stuff. Why is biological manipulation a better choice right now? Long story short, for prosthetics, we've been stuck on the same problem for years. Our bodies run by making the chemical reactions that sustain us statistically probable, but adding in metal or plastic or whatever throws a wrench in that in a way our bodies can't handle. When cells that start messing up and dying near the implants, the body treats it like an infection. So unless we start making prosthetics out of chemically inert materials, we're kind of stuck trying to build basically the body's ecosystem on top of all of these, which is obviously not an easy thing to do, or else we'd have done it by now. So how is biological manipulation doing any better? Well, consider the following actual experiments researchers have done. So first off, researchers wanted to identify what part of a mouse's brain controlled aggression. But stimulating the brain using electrodes was too imprecise. The shock would spread out too much. So what did these mad lads do? They took the protein our eyes use to react to light, strapped it to a tiny gate, and then engineered a virus that would go into the mice nerve cells and insert DNA, causing those cells to produce the light-activated gate and then put it in their own cell membranes. Basically, this made it so if you shined a light on it, this gate would open and it would cause all of the ions in the cell to equalize out, which is like what happens when your nerve fires. This is how you get a nerve to fire. And to be clear, using viruses to change DNA in mice and in flies and in plants is alarmingly common. I had no idea before I started doing this research how common this stuff was. It's basic lab stuff. It's kind of scary. So after they modified the nerve cells, they go in and surgically implant this fiber optic cable directly into the mice brain, right? And they put light through it. Within seconds, the mice would go nuts. They would attack anything in their cage. They would even, some of them, 40% of them would even attack inanimate gloves. They didn't even have to, make the glove move around, in which case, 90% of them attacked the glove, right? They were berserk. So that's pretty crazy, right? But what does that have to do with making new hearts or livers or whatever? So just think about how well we have to understand how cells work to be able to do what we just talked about. To be able to literally have a nerve cell, which would normally never make this protein, make it, and then put it in its cell wall, which, the process of even making it is kind of insane because this virus has to come in, it has to match with the perfect segment of DNA, it has to snip it open, insert this new DNA, and then seal those, and it has to do it right every time. It is insane that we can do this and that we can basically, mind control rats if we put a little LED in their brains and then use that to just make them go crazy. That's crazy. And if that's not enough for you though, the mad scientists of biology, they didn't stop there, right? We've modified the DNA of flies to grow legs where their antennas should be, just to prove that you can change one or two things and different body parts just come out where they're supposed to. And then they made flies grow a second set of eyes on their knees. Now they didn't make it grow the nerves to make those work, so it was just a completely non-functional set of eyes on their knees. But that's horror movie stuff. That's insane. And so that one's a bit clearer though, right? We can tell an embryo what to grow and where. We could basically have an embryo turn off everything but the liver grow part, right? And we could just make it do that. And if they have a genetic defect, we can just edit that out. I'm sure a lot of people have heard about CRISPR. We can just change the DNA to be what we need it to be. And since it's that person's DNA being used for the embryo, there's no possibility of rejection, right? We've custom grown the perfect organ for them. And like the only reason we probably aren't already doing this is because it's illegal to experiment on embryos past a certain point. So like we can't get them to grow this far because we're literally not allowed to. But so we already control biology so well that if like we had a moonshot type program and the government just said like by the end of the decade we're going to do this and we're going to make the legislation and the regulation for this to be fine by then, like I have no doubt we would make that deadline. And cells, like the crazy thing about cells is they're designed to do this, right? Like cells are made to reproduce. They're made to grow. Everybody does it once, you know? And that's not true for prosthetics. Like when we're talking about trying to build these tiny body ecosystems on the surface of all these materials to put in the body, but also have it like not get in the way of sending signals to and from nerves and not having nerves react to them in negative ways. We're talking about manufacturing stuff on the micrometer, the nanometer scale. And manufacturing stuff with that level of precision is really, really hard. Like, it's still not something we can do at a mass producible scale, and we'd have to be tailoring each one of these to a person anyways. So it's still custom for both of them, but in one of them, you basically just insert the original DNA from the donor in the first place, and it kind of just does the rest of it. Like, it's actually surprisingly little customization instead of having to, like, you know, custom put on the correct antigens on every part of the surface to have the body not reject something implanted into it, which is way harder. And like biological methods are kind of shockingly cheap. Like you ask people working in biology labs at the university, and they're all kind of doing it because it's not expensive. So yeah, that's why biological manipulation is scary real, and I have basically no doubt we'll be doing this stuff in my lifetime. I hope you all learned something, and if you're not exactly, you know, excited about what you've learned, maybe a little scared, but still impressed. So yeah, thanks for listening.